Okay, group. So this is our second ed puzzle for Unit 4, which is biochemistry and macromolecules. So let's go ahead and jump into it. It's going to be a pretty short ed puzzle today. All right, so kind of keeping up with our intro to chemistry terminology that we really need to know, especially for the biochem portion of this unit. Solutions and suspensions. This is how different substances interact with each other. So a solution, anytime that we're talking about a solution, we're talking about some substance being evenly distributed or evenly mixed in to another substance. Typically, when we talk about solutions, we need to refer to and identify a solute and a solvent. So the solute is the substance being dissolved. And in this picture, it would be the sugar or salt, whatever this may be, because once it comes into contact with the water, those large molecules of, let's call this salt, these large molecules of salt, where they're all really bonded together, they will get separated and then water will kind of encompass them, making salt water. The solvent is the substance in which the solute dissolves. So in this case, it would be the water because the water is the thing making the salt compound or the salt ionic compound separate into individual molecules. So you can add to your notes that we can call water, it's actually known as the universal solvent. That's a way for me to kind of remember solute versus solvent. Universal solvent. And I just remember it because water dissolves a lot of things. You can put a lot of stuff into water and it will separate those molecules and it will evenly dissolve into it. Now, on kind of the flip side of that, when you mix substances together and they don't evenly disperse, they don't evenly mix in with each other, we call that a suspension. So in this picture over here, we can see oil and water. The oil sits on top or it suspends on top. It's suspended in the air. And you can tell exactly where the oil starts and ends, and you can tell exactly where the water stops and ends, or starts and ends, excuse me. So why water is so important? We just mentioned on the last slide, it's the universal solvent. It interacts with anything that is ionic and anything that is polar. So you can add that as well. So anything ionic or polar, water will dissolve that. So anything that is covalent or nonpolar, the water cannot touch. But most of the substances that we ever try to put into contact with water, with the exception of like fats, they're all going to be ionic or polar. So that's why we call it the universal solvent. Water also has this thing called a high specific heat. And what this means is it takes a lot of energy to heat water up, and it takes a lot of energy being taken away to cool water down. This is why after you've been like boiling water, let's say you are boiling water to make mac and cheese, you pour the water for some reason into a separate container. The metal that your pot was made out of is going to cool down very quickly, but the water that was boiling that you put into maybe a glass container, it is going to stay hot for a really long time. And that's because it takes a lot of energy being taken away from the water to get it to cool down. Likewise, it takes a lot of energy given to that water to heat up. If you think, again, the same kind of concept, if you have water inside of a pot, you're trying to boil it, that metal pot is going to heat up a lot faster than the water inside of it, which is why when you're making food, you might still be able to touch the water inside the pot even after it's been on for a little bit because it takes a lot of energy to get it to boil. But once it starts to boil, it's going to stay hot for a long time. And that's really important for our cells because our cells are made out of 70% water. And as a result of that, it takes a lot of energy getting added to those cells to make them hotter, but it takes a lot of energy getting taken away to get them to be colder. So it's a really good, water is really good for homeostasis and maintaining those internal conditions. All right, acids, bases, and pH. So pH is just the way that we measure how acidic or how basic something is. So we measure it on a scale of 1 to 14. And on the next slide, there's a little bit more information about it. So, but 1 to 7 means that the substance, whatever it is going to be acidic. And then 7.0 is a perfect neutral. Anything above 7.0 to 14 is going to be considered basic. And how we measure these is what we look at is the breakup of hydrogen ions versus hydroxide ions. So in this diagram of this reaction, water can dissociate into one hydrogen ion in one hydroxide ion. So because they are equal to each other in terms of their power, so one is a positive one, one's a negative one, water after being disassociated it's a balanced perfect neutral. So it would be at a 7 on the pH scale. Acids, anything that is from that 0 to 7 or 6.99 or however you want to calculate that, anything 0 up to 7 is going to be have a high concentration of hydrogen ions. And because of this, the way that we can kind of measure that is we look at litmus paper, and using this, any of the blue litmus paper, if it's coming into contact with something acidic, will turn red. And it's because of those hydrogen ions hitting the substance that the litmus paper is made out of. Bases have a low hydrogen ion concentration. They actually have more hydroxide. So anytime you see a substance and it ends in an OH, so I'm trying to think of an example. Baking soda, for an example, is sodium hydroxide. It is made up of NaOH, and so because of that, it is a basic substance. So any pH that's over 7, so here, kind of from 7 all the way up to 14, this is going to be stuff that's basic. This is what we call alkalinity, alkaline, and that's because anything that is alkaline will have basic properties. Not basic like she's basic, but basic like the substance. Now, things that are basic, they're going to have like more of a bitter taste. Not that I want you to go around tasting things trying to figure out if they're acidic or basic, but things that you can consume, they're going to have more of like a bitter or like a salty taste to them. They are going to be very slippery. So like bleach, soap, things like that, they have that slipperiness to them. Whereas things that are acidic, those are going to be more sour, and they're going to be more like almost feel like watery in a sense. So you can look at the scale here and just see how many things we come into contact with that in a daily basis that they do have very strong acidic or basic properties. But because of what they're kind of interacting with as well, that's why, like, for example, orange juice is very acidic, but it doesn't eat through our skin because our skin has basic properties to it, which can neutralize each other. They kind of cancel each other out. All right, last little bit here, buffers. Buffers are going to be something that basically neutralizes an acid or a base. So they try to maintain a pH in or around 7 by changing the concentration of ions. So, for example, if we're doing a lab and we spill hydrochloric acid, can be a pretty strong acid, what we can do to help buffer that or neutralize it is we can pour baking soda on it. And so the baking soda with its NaOH, so I can kind of write this out here. So baking soda is NaOH. Hydrochloric acid is HCl. So as a result, what happens is the sodium hydroxide gets rid of its OH because it's coming into contact with the hydrochloric acid. The hydrochloric acid gets rid of its hydrogen. So then these two come together to make water. And then what's left is NaCl, which you know that as table salt. So when you take a strong acid and you mix it with a strong base, all you get is some sort of salt water. So, but buffers are going to play a really important role in maintaining homeostasis. If the pH of like a cell gets too high or it gets too low, a buffer can be introduced to that to try to get that to even out. So with a neutral pH, litmus paper does not change colors. Blue litmus will stay blue, red will stay red. But some biological buffers are bicarbonate, which sodium hydroxide would be a bicarbonate, and then phosphate, which is POH4 or something like that. PO4H. But anyways, so these are two things that you can introduce to a cell to try to help neutralize it being too acidic or too basic. So, all right, well, that is it for this ed puzzle. As always, if you have any questions, please let me know. Otherwise, I will see you in class for us to discuss and do some activities. See you then.